All right, you guys go ahead and get out a sheet of paper, and you'll take these notes and you'll turn them into me next class. So shouldn't make that long of a video. We're just going to go through a couple organelles. The first one is, as I said, probably the most important for life to exist, considering no life exists without ribosomes. Ribosomes make proteins from amino acids. So basically, there's all these amino acids are floating around kind of loosely in the cytoplasm. They're all out there. And what ends up happening is that a ribosome is the location where the actual, so here's, let's say, our nucleus, a mRNA and two parts of the ribosome come together, and then all the little amino acids, they get linked together in this long chain of amino acids to make our actually polypeptide sequence. Okay, so that's what a ribosome actually does. And their composite, what they're made out of, is RNA and proteins. This typically happens in the cytoplasm. There's two general classifications of ribosomes. They're what we call free ribosomes that are free floating in the cytoplasm themselves, and they are found in eukaryotes and prokaryotes. They're also attached ribosomes, and those are only found in eukaryotes, and they're attached to a membrane. So they might be attached to the nuclear membrane. There's also this thing we're going to learn about called the endoplasmic reticulum. They might be attached to that, but those are called attached, and those, once again, are only going to be found in eukaryotic organisms. They are the most common, and they can have as many as 15,000 in just a bacteria alone. So there's tons of them in a given cell, and I think they're pretty important. Here's an actual image of the ribosomes. Kind of see them zoomed in. So there's one. There's a bunch of them in here. That's what all these little dots represent. So those are all different ribosomes that you can kind of see. You can kind of see the DNA or the mRNA or the actual amino acid being synthesized off of that. The next one kind of goes along with that is called the endoplasmic reticulum. It's commonly referred to as the ER. Its definition is it's a network of interconnected, flattened, or tube-like structures. It is a transport system. Think of it as a subway or railroad system. There's two general classifications of endoplasmic reticulum. The first one is called our rough ER, and that is for when there are ribosomes attached to the ER itself. And then the reason why those are attached is those ribosomes are going to make proteins, and those proteins are going to be used to be transported either throughout the cell itself or to another part of the cell to actually be sent out the cell. If the ER does not have any ribosomes, we call it smooth ER. Okay. Sometimes, not always, the rough and the smooth ER are connected, and they're typically located near each other. So if you look at a cell, the nucleus, the rough ER is typically near the nucleus. There's our smooth ER, and then what you can see is, I want to make this a little bit bigger. So these are rough ER, and we can tell because there's ribosomes attached. Here's our rough ER, those are all ribosomes attached. Those are attached ribosomes, these would be free floating. Notice the smooth ER completely lacks that. Okay. I like this image of it. It kind of shows a couple things. Number one, the location. The location of the rough ER is near the nucleus because it needs to transport proteins maybe in the nucleus or throughout. So that's the rough ER. You can see those little ribosomes or little dots or studs on there. Then the smooth is this region right here, always found near the rough ER. So you have the rough ER is found near the nucleus. The smooth ER is found near the rough ER. Once again, all you're doing is really taking general notes. There's no specific questions or anything like that I have you complete right as of right now, just general notes over the video. Just another image of that. Notice from the diagram here, this is the rough ER. Okay, that's the rough ER. You can tell by two reasons. One, location near the nucleus. Two, the little dots right there represent the ribosomes. This region down here, that's going to refer to the smooth ER. Notice there's no ribosomes attached. This is an actual image I'm using a TEM microscope. The rough ER, a lot of times people look at this and they'll see stuff like this, these things here, and go, oh, those must be the ribosomes. The ribosomes are actually these little dots in here, the real tiny pepper-looking things. That makes this the rough ER, and then the smooth ER lacks those. So this is kind of the boundary for the rough ER. So smooth's on that side, rough is on that side. And once again, you can tell because of those for two reasons. Number one, you can tell because of the small dots. This is also where the nucleus would be at, and so that's another reason you can kind of tell. The Golgi bodies are another transport organelle in the cell, and their location is always near the ER. And a lot of times they do appear similar to the ERs because they're actually based or created via vesicles that break off from the ER. And I always think of the Golgi body as kind of like the UPS store, or if you go to Kinko's on 172nd and Dodge, that when you go to the Kinko store, you can do a lot of different things. You can ship things, right? You can package things. You can store things. Okay, you can receive packages. All of that can happen at one location. The Golgi body, it's going to do all that. It's going to ship things out of the cell. It's going to package things together. It's going to store. It's going to receive. So it is like the UPS store of the cell. Here you can actually see the Golgi body. These are little vesicles that are forming off of there, and they're going to be transported typically to the plasma membrane. There's two regions, a region that accepts packages, usually from the Golgi apparatus, from the ER, and then you can package it, it can label it where it needs to go, and then it'll send it out the other end. So this would be the receiving end, that is the shipping end. It receives from the ER and it can send it outside the cell. This is also often referred to as the Golgi apparatus. The mitochondria, when you were a little kid, or at least I was looking at my wife's fourth grade book one time, and they had a mitochondria, and I kid you not, it kind of looked like this. It looked like a kind of a peanut shaped, and there's little legs. For some reason, they decided to put a top hat on it, right? And they did this, and they kind of put arms up like it was flexing, and I'm running out of space, so kind of do it like that. And they said that the mitochondria is the powerhouse of the cell. Great. Awesome. Don't know why you'd want that as kind of your representation. The mitochondria, its job is that it's, number one, it's not always the same shape. It can kind of manipulate and change over time, but it is the site of aerobic respiration. Basically, it's where ATP is produced. As I mentioned before, the ribosome is the reason life exists. You need to have ribosomes. The major difference between eukaryotes and prokaryotes is the existence of mitochondria. Mitochondria are going to produce because of mitochondria, for one glucose molecule, it's going to produce a net of 36 ATP. Okay, without the mitochondria, so no mito, net, for the same amount of glucose, you only get 2 net ATP. So just think about that. 18 times more energy because of mitochondria. The reason why we and all of the eukaryotes are so advanced is because we have so much more energy available to us because of the mitochondria. Mitochondria is the site of aerobic respiration and provides us with all the energy that we need. There is an inner fold of the mitochondria called the cristae, and that cristae is it's responsible for increasing surface area to maximize the sites of aerobic respiration. Increased surface area means more spots that can actually do aerobic respiration. Mitochondria are found only in eukaryotic organisms, and they're able to get the energy out of your food. Once again, they're found in almost all organisms. There's a couple that don't, that are eukaryotes. They are considered the powerhouse of the cell, and really you can think of that way because they provide energy. So what type of cells would you expect? A lot of mitochondria, ones that need a lot of energy. Muscle cells, for example, your cardiac cells, those require tons and tons of energy. They're going to have a lot of mitochondria. Conversely, the reactions of this require ribosomes. So mitochondria will also be near ribosomes and have their own ribosomes in order to provide the proteins necessary for aerobic respiration. And there's an image of a mitochondria. There it is. Okay, these right here, you should kind of have this labeled, be able to draw something similar to this. That is the cristae, increases surface area, and then there are ribosomes that are actually embedded in the inner surface as well. The cristae increase surface area in more places. These ribosomes provide proteins for the reactions of cellular respiration. And then there's another image. Notice it can kind of move and flip and shape. And then down here kind of shows the increased surface area that exists in the mitochondria itself. I'm going to stop here. We'll talk, this is where I'll pick up next time, so you don't have to worry about this. Thanks for watching the video. Make sure you take those notes and bring them to class. That's all I got. Have a good day.